I'm trying to learn a new program, so this is actually my third attempt now at recording this video, trying to help you to better understand the steps of muscle contraction. The first video will cover excitation through excitation-contraction coupling, and the second video is going to focus on cross-bridge cycling. And this is something I usually do on the whiteboard in class, but because we're not able to meet in class, I'm not going to do that this time. Okay, so what I've drawn here is a somatic motor neuron with acetylcholine containing synaptic vesicles in the synaptic knob. All right, so we have the somatic motor neuron. Okay, so now I've labeled the synaptic knob and the acetylcholine containing synaptic vesicles indicated in green there. What I'm going to draw now is the sarcolemma, which you'll remember is the plasma membrane of skeletal muscle. Now I'm drawing the junctional folds of the motor end plate. So I've labeled the sarcolemma over here on the right just to indicate the plasma membrane of skeletal muscle. Here is the motor end plate. And here are the junctional folds. Now on these junctional folds, there is, trying to figure out how to change color here. There is a high density, and I'm going to draw these in blue hash marks, of acetylcholine receptors. These are turning out as dots. I'm trying to use a stylus to do the drawing. I do not have an Apple pen. Okay. So these blue hash marks right here are transmembrane proteins that represent the acetylcholine receptors. All right, there's one more thing I need to add that I have not yet drawn in. And we will use orange here to indicate voltage-gated calcium channels. These are transmembrane proteins that are found on the synaptic knob. Now, if you'll remember, we used the word action potential in class to indicate a traveling electrical signal. And what's going to happen is that the brain sends a signal down to the specific parts of the muscle that it wants to recruit. And that signal is transferred via electrical signals called action potentials that we will talk about in the next chapter. And those travel down the cell membrane of the somatic motor neuron. And I'm going to draw that. Here's a squiggly line showing, that's an arrow at the end, showing that that's traveling down the sarcolemma. And eventually it gets down here to the voltage-gated calcium channels. And that's going to cause these voltage-gated calcium channels to open because it's an electrical signal. It's a change in voltage. Now, calcium is higher in the extracellular fluid than it is in the intracellular fluid. So when we open those voltage-gated calcium channels, calcium is going to come in to the cell, into the synaptic knob. Now, if you want to write out steps, I would write something like the action potential travels down the somatic motor neuron and opens voltage-gated calcium channels on the synaptic knob as step one. As step two, what the calcium does to the synaptic knob, it causes exocytosis of those synaptic vesicles that contain acetylcholine. And so when they undergo exocytosis, of course, they move to the synaptic knob, to the membrane of the synaptic knob, and then they release the acetylcholine out in the synaptic cleft. And I'm just going to indicate the synaptic cleft here with purple. Right here's the synaptic cleft here, just so you can see where that is. But that calcium now, and I'm going to indicate this in, no, I'm sorry, not the calcium, but rather the acetylcholine, which we've indicated in green now because it's released, is found in a high concentration out here in the synaptic cleft. That's where it just got dumped. And it diffuses across and binds to the blue acetylcholine receptor. So if we want to look at that in kind of a microcosm here, if we blow up the picture, here's the junctional folds down here. Here's the acetylcholine receptor on those junctional folds. And here is the acetylcholine in the synaptic cleft. And that acetylcholine comes over and binds to the acetylcholine receptor. Now, I'm going to erase this and start a new video showing what happens, and then we'll continue from there. Okay, once again, here are the junctional folds. Here is the acetylcholine receptor. I'm going to draw it kind of blocky this time. Just to remember, there's a high density of these on the junctional folds. And acetylcholine has come and bound to each acetylcholine receptor. And there's 2 acetylcholines that actually bind to each one, so I'm trying to indicate that, although it's difficult here. And what that does, the acetylcholine receptor, remember, we talked about in the lecture, is an ion channel. And so the ion in question here, I'm going to put in brown, is sodium, which we've talked about in class is higher outside the cell. Okay. And the first thing that happens is this channel opens and sodium rushes into the cell because it's so much higher outside the cell. It's coming through all of these receptors. And so that sodium comes through the receptors, increasing the intracellular sodium concentration. Because sodium is positively charged, that changes the charge now across the cell membrane. Now these, and it's really only a brief change because potassium is higher inside the cell, and right after the sodium rushes in, the potassium rushes back out and changes the charge difference back again because it has that positive charge. I don't consider that the most important part, but that's how membrane potential is essentially reestablished at this point. But what I want to really focus on is this increase in intracellular sodium. Okay? So now let's back up just a little bit. All right, so now I'm going to start with the somatic motor neuron up top with the synaptic knob right here. Here's the sarcolemma. Here's the motor end plate with the junctional folds. All right. One thing I should have drawn in here and did not draw in right now. So let's draw in T-tubules. Remember, these are deep evaginations to the interior of the myofiber. All right, and I just want to make sure that we draw those in there because we will use those in a few minutes. Now, before we go back to where we were, I also want to just cover a little additional information. Remember, on either side of a T-tubule, you have the terminal cisternae of the junctional, of the sarcoplasmic reticulum, the SR. And I'm going to draw that here in gray because I don't want to have too colorful for it. So we're just going to draw it like that. And I'm not going to draw any more of that, but remember the SR has other things that come out and kind of network, and I'm going to draw one on here on either side. I'll just draw another one right here. So that's the SR. All right, so I've labeled those there. And if you'll remember, calcium, and let's do calcium in green. I'm not going to write out Ca for calcium. I'm just going to put dots. Calcium is very high inside the sarcoplasmic reticulum, particularly in the terminal cisternae. Okay. And if you remember, we talked in the lecture, it also binds to a protein called calsequestrin, which allows it to store even more calcium inside the SR. I'm not going to draw that part in the video. All right, so that green indicates calcium. Okay. Now we're going to draw sarcomeres. So here I'm going to draw a Z disc. And here's another Z disc. And over here to the right, there'd be another Z disc. Over here to the left, there'd be yet another Z disc. And between these, we have, I'm just going to draw this all in black. I'm not going to label these parts, by the way. So those are the Z discs. Here's the thick filament. Remember, the thick filament is made of myosin. So this is the thick filament. The myosin heads that kind of stick out off the thick filament, there's some myosin heads. We'll draw those just as some little lines there. So those are the myosin heads. We have the thin filaments. I'm sorry, let's go and draw the thin filaments first. The thin filaments attach directly to the Z disc and come out and overlap where there are myosin heads. And again, this is a two-dimensional rendering. Really, the thin filaments, there would be 6 of them surrounding each myosin. And in my drawing here, these are touching. In reality, they would not really be touching when you're in a relaxed state. So those are the thin filaments made of actin. One more protein I'm going to draw in here, and that's the protein titin. You can see the thick filament has nothing right now to hold it in place. But the protein titin, which we said was a springy protein, so I'm trying to draw a spring there, attaches the thick filament to the Z disc. Okay. So there we have it. Now on the other side, there's more thin filaments going this direction. There are more thick filaments. I'll just draw it around there since this is not three-dimensional. There's more going this direction. I won't draw in all the details. That's just to remind you that these sarcomeres are found all the way down the myofiber. There's not just one sitting in the middle. They're found all the way down. That's why we have the light, dark, light, dark, light, dark pattern, because of the A band, the I band, the A band, the I band that appear all the way down. All right, so now I'm going to erase those back out because I don't want those to be there in my final picture. We need the space for some other things. Okay. So there's our sarcomere. Now what we're going to do is we're going to go back up to the neuromuscular junction right here. And we're going to look at what happens. Remember, we had ended in the previous part of the video where sodium had just rushed in to the cell, changing the voltage. Right, now importantly, that sodium just doesn't sit there. It eventually gets pumped back out. Remember, the sodium-potassium pumper, sodium-potassium ATPase is always active, and we'll pump the sodium back out again, pump the potassium back in, and we'll regulate everything. But briefly, this sodium can diffuse around in the different direction. And just over to the side is a neighbor that we'll draw in green. And this is a type of a transmembrane protein that is found all the way down the sarcolemma, including down into the T-tubules. Okay. And it goes the other direction as well. I'm not going to draw it all the way the other direction, but it does the other direction as well. This particular protein is a voltage-gated sodium channel. And we have the sodium diffusing right here. Okay? So that sodium just diffused, and again, think three-dimensionally, as it diffuses around, sodium has a positive charge, which changes the voltage across the membrane and opens the voltage-gated sodium channel. Okay? So that's where we are right here. Now, sodium, we've already seen, is high outside the cell. Well, if sodium is high outside the cell, and we'll keep drawing sodium in green here. Now I'm just going to draw it, though, as green dots. If sodium is high outside the cell, and I just open a voltage-gated sodium channel, that sodium is going to come in. Well, that would be the end of things, except now we have more sodium coming in, which changes the voltage again. And as that sodium diffuses down inside the cell, it opens the next voltage-gated sodium channel. And sodium comes in again. That sodium will open the next voltage-gated sodium channel, and sodium will come in. Sorry, I'm having a hard time drawing this here. So the sodium comes in, opens the next one, sodium comes in. So pretty soon you have the sodium coming in all the way down the sarcolemma, down the T-tubules as well. So sodium now has successively opened each voltage-gated sodium channel. So when we talk about excitation, this is excitation. We took the signal from the nerve, transferred it across the synaptic cleft via a neurotransmitter, acetylcholine, and now we have our own action potential or traveling electrical signal that's happening all the way down the sarcolemma, opening all the voltage-gated sodium channels, including those in the T-tubule. Now, let's look what happens in the T-tubule. I'm going to need to use some different colors here, and so I'll use green for both sodium and calcium. But remember, the green inside the terminal cisterna of the SR is calcium. Right? In brown here, I'm going to draw a protein. And we briefly talked about this in class, and I'm just going to draw it like this. And these proteins, I'm just going to draw it right here. But this brown protein, this is present all the way down the T-tubules. So I'm going over to the left. I'm going to draw it over here. But it gets too busy if I mark everything up. So this brown protein is the dihydropyridine receptor, and it's going to act as the voltage sensor, so that as the action potential comes down the T-tubule, as those sodium channels are opened, the dihydropyridine receptor will get activated. Now that dihydropyridine receptor is linked to another receptor that we will draw here in yellow. So again, these are linked to these yellow receptors. I'm drawing some here on the opposite side. So all of the dihydropyridine receptors are linked to this yellow protein. This yellow protein is the ryanodine receptor. The ryanodine receptor is a calcium channel. Now these two proteins work together, so the dihydropyridine receptor senses the change in voltage. It interacts with the ryanodine receptor and opens the calcium channel. So remember, there's a lot of calcium inside the sarcoplasmic reticulum. So when the ryanodine receptor is opened, there's a huge influx of calcium into the interior of the muscle fiber. Now, as far as the details go, the important parts to remember is that the calcium channel on the sarcoplasmic reticulum is regulated by the voltage that comes down the T-tubule. This is why the arrangement of the triad, remember terminal cisterna, T-tubule, terminal cisterna, that triad arrangement is important because now when the electrical signal comes deep inside the muscle through the T-tubule, it is able to open the calcium channels on the sarcoplasmic reticulum. And calcium now rushes out into the interior of the muscle fiber. And remember that calcium, I'm not going to write these steps out, but calcium binds to troponin, and troponin moves tropomyosin off of the active site on actin so that myosin can interact with it. We will cover that. We won't draw that out in another video, but we will draw the steps of what happens after that. So again, if you want to go back to steps, we started with the action potential travels down the somatic motor neuron and opens voltage-gated calcium channels on the synaptic knob. Calcium moves into the synaptic knob and causes exocytosis of the acetylcholine-filled synaptic vesicles. The acetylcholine then is exocytosed into the synaptic cleft, where it diffuses across and binds to the acetylcholine receptor. It allows, the acetylcholine receptor is an ion channel. It allows sodium to come into the cell, changes the voltage. The sodium diffuses to the neighboring areas around and opens voltage-gated sodium channels. The sodium comes in, and now you have an action potential that travels all the way down the membrane because of the successive opening of voltage-gated sodium channels, including down into the T-tubule, where the voltage is sensed by the dihydropyridine receptor, which causes opening of the ryanodine receptor, which is on the terminal cisternae of the sarcoplasmic reticulum. It allows calcium efflux into the sarcoplasm of the myofiber. And then that calcium binds to troponin, which moves tropomyosin off of the active site on actin. And now, because the myosin heads are interacting with actin, it pulls them. What's going to happen is it's going to pull them from the position it is right now, where you have the thick and thin filaments arranged. I'll draw this next part in brown because it pulls them. What's going to happen is those Z discs get a little bit closer together. The thick filaments don't change length. The thin filaments don't change length. But those thin filaments are pulled inward, pulls on the Z disc. And when that happens millions of times across and down the length of a myofiber, it causes shortening of the muscle.